Hi, I'm Jonathan Retarth, class of 2007, coming to you live from the campus of the University of Notre Dame. Welcome to our first Join the Fight webinar in partnership with Notre Dame's award-winning What Would You Fight For series and presented by Proud to Be ND. Today, we're joined by two leaders in the field of precision medicine. Professor Paul Bohn. So we'll do this again. And Professor Paul Bohn is the author, Arthur J. Schmidt Professor of Chemical and Biomolecular Engineering. He is a Notre Dame class of 1977 and went on to earn a doctorate at the University of Wisconsin-Madison. He's been on the faculty here at Notre Dame since 2006, is the author or co-author of over 225 academic publications, and has had 6 patents issued from his research in the fields of analytical nanofluidics, low-dimensional analytical electronics and photonics, and correlated imaging. Professor Norman Dovichi is the Grace Rubley Professor of Chemistry and Biochemistry. His work and the work of the Dovichi Research Group was critical in developing the instrumentation used in the Human Genome Project, and his team continues to study protein expression and metabolism pathways in single cells, which are necessary in understanding health and disease. Welcome to both of you gentlemen. Thank you so much for joining us today. We're so happy to have each of you to join the conversation as we pick up from this week's amazing What Would You Fight For piece, which we'll be showing in just a second. Throughout our session today, we're going to be accepting questions. Please use either the Google Hangout sidebar for those of you viewing us on Google+. You can also email us, proudtobenotredame @gmail.com, or you can post on Twitter using the hashtag ProudToBeND, and we'll pick up your questions. We'll feed them through to Professors Bohn and Dovichi as we move along. For those of you who might not have seen it or like to see it again, we're now going to queue up this week's What Are You Fighting For video. It involves the field of work of sepsis of these two gentlemen. It showcases the early sepsis detection and targeted treatment work of these two men as leaders in the field of precision medicine. So we'll queue this up right now, and you can check out this week's Fighting For video.
I was a doctor in the emergency room when an 11-month-old girl named Janice arrived with burns over 75% of her body. Her chances of survival were very slim, but after multiple operations, she pulled through. 4 weeks after she arrived, we helped her celebrate her first birthday. The next day, she died in my arms. Her body had succumbed to sepsis.
Sepsis is a severe reaction to infection, causing 40% of all hospital deaths in America. At the University of Notre Dame's Advanced Diagnostics and Therapeutics Initiative, Professors Norm Dovichi and Paul Bohn are developing a molecular test that will identify sepsis at its earliest stages.
Sepsis can be very difficult to diagnose because the early symptoms are often mistaken for the flu or common cold. Our tests will give medical professionals the information necessary to detect the onset of sepsis, which, if caught early, is very treatable.
Norm Dovichi helped map the human genome, and together at Notre Dame, we're taking that research to the next level, using precision medicine to make real advances in healthcare.
In collaboration with the Feinstein Institute, Notre Dame's researchers will work directly with physicians to design diagnostic solutions for complex health challenges like sepsis.
Having studied sepsis for more than 30 years, I'm incredibly excited about Notre Dame's research. I know the potential this has to save millions of lives.
The University of Notre Dame asks, what would you fight for? Fighting for precision medicine.
We are the Fighting Irish.
That you have a presentation that you'd like to share as part of today's webinar as well. Would you like to get started with that?
Yes, I would. I'd be happy to, Jonathan. Thanks very much for joining us this afternoon. Norm and I are delighted to be with you. We want to talk a little bit more in depth and flesh out some of the ideas that were presented so beautifully in that video. Just a few short slides to kind of get us oriented today. So to begin with, going to the second slide, I think you can see in this graphic from the journal Nature that if we look at some of the most common and popular medicines in the United States, the ability to actually impact positively the health status of the patient population varies from not very good to really horrible. And in part, this can be thought of as the interaction of the specific medication with a specific individual, and it's just a problem that we have not yet wrapped our arms around. So in this case, all of the blue individuals are the ones who are being helped by the medication in a given case, and all the red ones are the ones who are not being helped on average by that medication. Now, on the next slide, we really make the point that precision medicine is an idea that is already garnering a lot of attention all across the country and indeed worldwide. In fact, the President in his State of the Union address in January directed the National Institutes of Health to commit over $200 million in its program to a broad effort to use molecular markers in order to direct therapies in order to specifically improve the prospects for medication helping individual patients. So precision medicine can be thought of as the problem of how to deliver the right treatment to the right person, but not just at any time. That treatment has to be delivered in a way that is appropriate in time and space so that the treatment has its optimum effect for that particular patient. And understanding how to get this right is really at the heart of what we're trying to do at Notre Dame. So as one example that I think has gotten a lot of press recently, the breast cancer drug trastuzumab, or Herceptin, is a drug that was developed in order to be specifically positive for women who carry the HER2 genetic profile, and it's shown to be to have improved prognosis for those women. Now, why Notre Dame? Well, we believe that we have assembled one of the best bioanalytical groups. It's a group that's growing quickly. We've gone essentially from ground zero to a very vibrant group of 7 full-time faculty in just a few short years. And the university has invested both in strategic research initiatives to support our work as well as in faculty hiring initiatives. So as we look to the future, we're very excited and optimistic about our ability to bring even more and better and smarter and additional talent to the problem. In addition, we're holistic. So if you look around the country and you delve into precision medicine initiatives, I think what you'll see is that by and large, those initiatives are looking at genetic information, and while valuable, genetic information can only tell you part of the problem or give you part of the picture because it stops at a particular point, and that is the point at which the genetic information is translated, transcribed and translated into the molecules that actually do the work of the cell in our bodies, those being the proteins. And then, of course, when the proteins do their work, they produce product molecules, the metabolites. And finally, all of that occurs within an environment. So as we think about how to move beyond genomic information here at Notre Dame, together with my colleague Norm Dovichi and his students, they're looking at a variety of cutting-edge approaches to obtaining proteomic information and metabolomic information, which goes well beyond the genomic information that's being used by the scientific body at large. We're excited to have a number of partners in this effort. The Feinstein Institute, I think everybody was really quite taken with Kevin Tracy's description of just how important sepsis is, both from a medical perspective, but also from, I think, from a very real and personal perspective for him. And we have gone from there and developed collaborations with a number of other entities that you can see here on this slide. St. Joe and Memorial, of course, are local hospitals here in South Bend, Indiana, and then you can see others around the country, both clinics, hospitals, and indeed private companies as well. So what sets this program apart? We are, to a very, very large degree, use-inspired. We sit down with clinicians and we talk with them about their problems. Our initial focus on sepsis was entirely driven by a set of conversations both locally and with our colleagues at the Feinstein Institute about the problems of sepsis and wound healing. We seek high-risk, high-reward projects. We want to make a difference in people's lives, and that means that some fraction of what we do is unfortunately going to fail in the sense of not producing a fieldable product, but still succeeding in the sense of helping to train students and bringing focus on this important medical problem. And finally, a central tenet of what we're doing here at Notre Dame is to bring together people from across disciplines, from chemistry, chemical engineering, biology, electrical engineering, etc., in order to bring a holistic approach to these very difficult problems. We're very focused as well on pushing what we develop back out into the community, and so as part of that, we are focusing a lot of our effort on as quickly as we can commercializing the ideas and helping launch small companies which can then take the research advances that we make here in the laboratories on campus and move them out into the larger population at large. So with that, I'm going to conclude my comments with these few brief slides, and we'll open it up to Jonathan and to the questions that you might have. And so once again, thanks for being with us.
Jonathan's muted again.
Okay. Sorry, I was. Thank you guys again for joining us. Let's start to get the questions. If you have questions, proudtobend@gmail.com. The hashtag proudtobend. You can reach us through that as well with questions. So, Professor Dovichi, the ad mentions your contribution to the Human Genome Project. How does that work inform this project and the work that you do now?
The experience in developing technology to sequence the human genome provides a strong basis for development of additional technologies and new technologies. But I think the most important thing from that experience is the importance of interacting with real-world problems. And that's where our focus on talking to the people at our local medical centers and also in the Feinstein and other locations around the country in order to identify important problems with which we can focus the attention of the wonderful team that's been assembled at Notre Dame is really quite important.
Terrific. I was really moved by Dr. Tracy's story. How did either one of you end up first meeting with him or working with him? How did this relationship kind of come together?
Well, I'll take that one, Jonathan. I was presenting the work that we had just started in the Precision Medicine Initiative about a year and a half ago to a group that was organized around the Cleveland Clinic Alliance, and the Feinstein Medical Research Institute is part of that alliance. So we met Kevin, who practically jumped out of his chair when he saw what we were doing here at Notre Dame. They have a very large research program, not just one or two investigators, but a whole set of principal investigators there at Feinstein who are focused on the biomedical aspects of sepsis and the molecular biology, cell biology related to the syndrome that's referred to as sepsis. And when they heard about the kinds of technologies that we were developing in Norm's group and other groups here at Notre Dame, Kevin was exceptionally excited and approached us, and we then began a series of conversations. We've exchanged visits with each other, and now have a series of projects between investigators here at Notre Dame on the basic science side, and then investigators at the Feinstein Institute in New York who are clinicians and are doing biomedical work.
Terrific. I mean, as a follow-up to that, you showed the big kind of list of hospitals and research centers that you work with. Can you say more about how departments like yours at Notre Dame work with physicians and hospitals, and of all the different areas that you could treat, or of all the different needs that hospitals have in the research space, how does the university or your departments or you individually as researchers, how do you choose which problems to tackle?
Well, that's a great question, Jonathan. We first we sat down and we didn't know which problems to tackle. We really took a look at things, and it was clear to us that we needed to be informed by the real-world problems of clinicians who are out on the front lines treating patients every day. And so we went and sat down with our local clinicians, both at St. Joe and at Memorial, and just talked to them about what their most difficult problems were. And sepsis and wound management really came to the fore in those discussions. So identifying sepsis really came out of a set of very frank and open conversations with a group of local clinicians. Now, in terms of what they can bring to the table and why it's important to have collaborative work across the basic science, clinical science divide, is the kinds of samples that we would be able to work on as a group of university investigators here at Notre Dame would be quite artificial without the connection to clinicians. So they can provide us with human-derived, patient-derived samples in a variety of ways that we would never be able to access on our own. So it's this connection, a collaboration between basic science, but connected in a very deep way with the real problems of clinicians that's really critical in this relationship.
Terrific. Professor Dovichi, what's the environment like out there for researchers who are also inventors, the commercialization of ideas? It was mentioned briefly in your piece, but obviously the idea of technology transfer is key to so much of the work that's being done at the university level. How does that influence your work? How has it, certainly with the Human Genome Project, influenced your work? And how does it guide your decision-making when it comes to new projects and your thinking around the future of such projects?
That's a complicated set of questions that you've answered or asked. The University of Notre Dame has some outstanding technology transfer officers working here. We work with industry on a number of projects, and we file patent disclosures on a number of our technologies. And we've been successful, fortunately, in licensing several of these to a number of companies. So having access to strong technology transfer personnel, people with good experience, is of course invaluable in guiding the technology development. It's very difficult to predict what's going to be successful and what's not going to be successful. So one needs to plant many seeds and see which of those will grow into fruitful trees. And in this case, working with our colleagues and collaborators at the local medical centers and at the Feinstein, it will be interesting to see how that technology comes to pass, and if we are successful in developing useful biomarkers, how the commercialization of that technology will proceed. That's very much up in the air and an exciting opportunity to learn.
Great. Once again, we're so happy to be talking with Professors Bohn and Dovichi, jumping off of this week's fantastic What Would You Fight For series on sepsis treatment and precision medicine. We encourage you to keep submitting questions. We've been getting a lot of great ones. If you're in Google Hangouts right now, you can submit it right in the Q&A module. If you're viewing us on Proud to Be ND on YouTube, you can submit via proudtobenotherdame@gmail.com, and we're checking that email inbox right now. Or you can submit your questions using the hashtag ProudToBeND on Twitter. So please keep them coming. We were talking about the importance of tech transfer and having those great kind of resources so that new great ideas can be commercialized in the university space. Professor Bohn, how does that impact some of the work that you do independently, and how has that been a factor for you in your career?
Well, it's really important to be able to have a good impedance match with the outside world. And we work with a particular small company, Hasiri Diagnostics, which has licensed some of the technology that we've developed in our laboratory here for the identification of specific strains of bacteria. And there, the end goal is a little bit different, but some of the underlying technology is very similar to what we use in our sepsis-related work. But the critical thing about having a connection with a company is that what it allows us to do is, at the university, we can do what we do well. We can work with our students. We can concentrate on the fundamental development of the science and technology underlying the treatments, the diagnostics, the therapeutics that we want to push out into the field. whereas our colleagues in the company in Hasiri Diagnostics, because they have medical training, they have experience in the pharmaceutical industry and the medical device industry, they can navigate all of the complicated regulatory and approval processes that are required in order to rapidly move the research advances that we make from the laboratory into the field. So it's this translational component that is so important to have good collaborators at companies as well as collaborators in hospitals and clinics.
Great, great. Well, I mean, certainly, you know, the need to have that connection is vital. I think also vital is a connection between researchers like yourself and the students at the university level. Professor Dovichi, can you talk to us a little bit about the influence of being able to work with students both at the graduate and undergraduate levels and how that's impacted your time here at Notre Dame?
Well, it's kind of what makes me get up in the morning. It's a great fun, great delight to work with these smart young people, see them make the conversion from being a student to being a scientist, being able to think independently, work independently. And it's also a great delight to see them succeed in their careers afterwards. I just came back from a conference in Beijing where I ran into a dozen alumni from my group, and it's just absolutely delightful to see these people succeeding. It's a source of great pride and pleasure.
How are they involved on, I guess, a day-to-day basis? So students are either they're taking classes, they're experiencing other parts of life at Notre Dame, either at the graduate level or undergraduate level. What are they doing for you each day? What does that immersive kind of an experience look like on a daily basis? And certainly open to both of you.
Well, the students it depends, of course, if graduate and undergraduate students. The graduate students very quickly move through the coursework and are devoting a very large fraction of their time to research. And they will be spending time generating samples, doing the chemical analysis to generate data, and then spending enormous amounts of time in front of the computer trying to interpret and understand the data. And then finally, if it's most important, is writing up the results into publications and patent applications to share with the wider community. The undergraduates are a bit different in that their lives are more complicated. They have full course loads, they have busy social lives, but they will be spending many hours a week working with graduate students, working on their own in order to move the whole enterprise along. And hopefully they're picking up and learning along the way how to think independently, how to work independently, how to interpret data, how to deal with success and failure in the laboratory, and then finally how to share the results with the wider community.
Thanks again to everybody for providing such great questions. We have a new one from Jenna who asks, where do you see the biggest impact for precision medicine? Where is it making the biggest impact, and for what type of patient?
Well, let me take a swipe at it, and I guess what I would do is I would start with where we are today. And our current implementation of precision medicine, as I indicated in my slides at the beginning, is to use genetic information, which, let me remind you that Norm Dovichi was integral in making it possible to even have that information, but using genetic information to essentially direct a relatively small number of therapies to specific patient populations. So we called out the HER2 gene in breast cancer as a specific example where there was a increased efficacy of a specific medication based on the presence of that genetic marker. And so currently, that's really where the technology lies. Now, what we see Norm and others here at Notre Dame doing and where we want to be, in 5 or 10 years is broadening that to the point where we're now using a much broader spectrum of molecular markers that can be acquired through blood or sputum or urine or whatever mechanism is appropriate, and then used to not only specify a particular drug, but as the course of the illness or the course of the treatment progresses, to feed back information on a real-time basis to a clinician to assess whether that drug is having the the intended effect or not. So it's almost like the difference between, if you love baseball like I do, setting your lineup card at the beginning and letting it go. That's what we do now. Versus the manager substituting players during the course of the game and changing the way in which individual therapies are managed by the clinician. So it's the power of this new information is really to put a tool in the hands of clinicians that they can use to better manage the course of treatment for individual patients.
Right, and following up on that, Professor Bohn, precision medicine has been getting kind of more talk in the media, both scientific and national media. How do the efforts that are happening here at Notre Dame, how do those align with kind of the national conversation on precision medicine and how it's kind of transforming the fields that you work in?
Well, we hope that we're on the leading edge of that conversation. I think that there are really terrific groups of people who have the right genetic capabilities and the right bioinformatics capabilities to really do a great job of using genetic information for precision medicine. What we want to do that is different and distinct here at Notre Dame is to provide new tools that go well beyond the acquisition of genetic information. So the example I always give to people is that having genetic level information is like having a blueprint for a house. Yeah, you sort of know what the house is going to look like, but having the protein and metabolite information is knowing what color the paint on the walls is, where the rugs are, what kind of appliances you got. And so the blueprint gives you some information, but it's the rest of that information that really distinguishes the house from the blueprint that you started with. So where we are at Notre Dame is very much in providing the tools that will allow clinicians to have that increased level of information about how their therapies are proceeding.
Let me amplify that a bit. Genetic information is proving very valuable for improving therapy for cancer. And cancer is a disease of the genome. But wide swath of other diseases, heart disease, sepsis, as we were talked about in the video, have at best a very weak genetic component associated with them. And instead we have to be a bit more sophisticated in our approach. The President's effort, NIH's effort, in precision medicine is primarily focused on using genetic information for characterizing cancer and improving cancer therapy. And we wish to have a bit more subtle and a bit more holistic view, looking at a wide swath of biological molecules obtained from the patient and use those to guide therapy.
Thank you guys, both. Stuart, again, thank you so much for everyone sending in questions, either through the Google Hangouts tab, through sending an email to proudtobenotre-dame@gmail.com, or by hitting us up on Twitter using the hashtag ProudToBeND. Stuart asks, what is the state of research now for finding new biological markers out there?
It's a really tough problem. There's been a lot of effort trying to develop markers for early diagnosis of cancer based on specific proteins present within the bloodstream, and those have been proven to be very, very challenging to validate. We are not so much interested in early diagnostics, not particularly interested in detecting diseases early. That's a very, very challenging project. But instead what we're interested in doing is guiding therapy, and they're working with clinicians. The real key bottleneck and the real key step is obtaining samples from patients in order to do prospective and retrospective studies. So obtaining samples from patients, serum, for example, and good clinical information in terms of the characteristic of the disease, and then the therapies that have been used, and then the outcome of the disease is real key. So this is a very long-term, very challenging set of projects. We believe by focusing on prognostics and identifying ideal care for a patient who does have a disease, we will have a greater impact, at least in the shorter term, compared to the efforts to develop early diagnostics.
Great. Well, a question that we just received from Andy actually flows right into that in terms of, speaking to the importance of finding new therapies or finding which people respond to the current treatments that are out there. So how do you kind of see the prioritization of that shaking out?
Well, Paul showed us a slide in his presentation, his second slide, which highlighted the small percentage of the population that responds to any particular therapy. So he showed the drug, I don't have my glasses on, pardon me, Nexum, which is treating heartburn, and perhaps 1 out of 20 patients will benefit from that therapy. And the only way you can discover which of those patients are going to benefit from therapy is to do the experiments, is to do the treatments. And that, of course, is a long-term and expensive project.
Right. Thanks again for everyone sending in such great questions. Larry asks next, will insurance agencies pay for the tests that make precision medicine possible? So looking at the end users, looking at the patients, looking at the overall kind of healthcare system and how finances factor into that. For precision medicine and its research findings specifically and the products that come to market, how does the kind of end payer and how do we've also received questions. Amber asks, how do pharmaceutical and biotech companies reach into that? So the commercialization of all of it, how complicated is that in today's healthcare system that we have in this country and around the world?
Well, I'll take a swipe at that question. The thing I would say is, first of all, in order for the insurance companies to really latch onto this, what they have to be convinced of is that there's a real benefit, financial benefit, again, in terms of increasing success of treatment, as Norm has talked about just recently here. And what I would emphasize is that here at Notre Dame, we're working on a variety of different technologies that range all the way from very, very sophisticated mass spectrometry and separation technologies, such as Norm's students are working on in their laboratory, that might be associated with high unit costs per test, all the way down all the way down to very, very inexpensive tests that can be administered with cheap materials at the bedside to obtain answers essentially by nurses in real time without waiting for a sample to go from the patient to the clinical laboratory and then back, and then for that information to flow back to the clinician and ultimately result in treatment. So we envision a wide variety of different levels of cost and sophistication for the tests that we're developing. And we can't do everything, but we do want to hit various parts of that spectrum. So we believe, again, that our corporate partners, the companies that are licensing the technology, will be really important players in developing the protocols and then also the preliminary data that can be used to convince the payers to actually include this as part of their insurance portfolios.
Great. Professor Dovichi, anything to add to that?
Paul, of course, nailed hit the nail on the head. And just to summarize it, the insurers will take advantage of precision medicine if it saves them money. If you are treating 20 patients and only one of them is benefiting, then the insurance company is wasting money on those other 19 patients. If we were able to identify those patients and only those patients who will benefit from the therapy, then the insurance companies should rapidly embrace this new technology.
Terrific. We've gotten, again, great questions from everyone that's tuning in and joining us. Thank you again so much for joining us in this first ever Join the Fight webinar presented by the award-winning What Would You Fight For series and proud to be ND here at the University of Notre Dame. Eleanor asks about impact and about timelines that you guys work with. A lot of times the research that you're doing can take years and decades to come to fruition, especially in actually reaching the market. Would this impact her 7-year-old daughter during her lifetime, the kind of research that's being done on certain things now? I know it really depends on the project, but what can you say about the overall timeline that's necessary and what kind of challenges you face when it comes to bringing things to market?
Well, again, I would point to Eleanor and say, you know, we have some things that we very much hope are going to be out there in the marketplace in a couple of years. We want to focus on ideas and approaches across the whole spectrum so that we are working both on long-range research that will ultimately impact people in 10 or 15 years, but then much shorter-range projects as well. I think Eleanor's 7-year-old daughter will be very likely to benefit from precision medicine developed and precision medicine targeted medications by the time she's an adult. I heard an interesting radio interview with Mark Cuban, who Mark is a businessman, as I think many of you know, and also a sports entrepreneur, and he was being asked this question within the context of his sports enterprises, and he was asked what he thought the most dramatic change would be in the American sporting landscape over the next couple of decades, and he went immediately to the idea of precision medicine and said he thought that if we were talking a decade or two from now, that there would be no longer treatments of athletes based on epidemiological information about treatment protocols and efficacies, and everyone would be treated with a specific precision medicine-derived molecular marker. I guess I'm putting words in his mouth, but that's what he meant anyway. A specific marker that would basically direct that particular patient to the right kind of therapy, whether it be a drug of some sort or whether it be surgical intervention or physical therapy or what have you. But basically, that was what he saw as the biggest change coming in the American sports landscape. So I think to wrap all of that together, I would say that change is going to start to happen very rapidly, and I suspect that Eleanor's daughter will certainly benefit from this by the time she's an adult.
Great, great. And yeah, actually, just to follow up on that, Lauren Gibson has sent in a question, very similar in terms of targeting specific populations. Clearly, the Fighting for ad focused on an infant with sepsis, but is the goal really to find biomarkers or therapies that will eventually be able to be tailored to every individual, or are there certain large groups of audiences or populations that are easier to work with when it comes to this research, or that you feel like you're closer with some populations than others?
Well, I think that's a great question. What I would say is that we know already that a disease is not a monolithic phenomenon. A disease impacts different ethnic groups, impacts different age population cohorts in different ways. And I could point to sepsis in particular. I had a very heartwarming email over the weekend from a gentleman on the faculty here at Notre Dame, and he had contracted sepsis at a hospital stay, and at his age, he was in his 80s. They gave him a 20% chance of living. I'm delighted to say, in fact, that he's well and he's back out and writing and publishing and working with students again. But in his case, he was in a high-risk portion of the patient population because of a variety of factors which would compromise his innate ability to fight off infection. So as we look at different ways to apply these tests, we're looking not just at applying them in a broad way, but also to apply them intelligently according to age, ethnicity, and other risk factors that may indicate high-risk status for a certain individual.
Right, right. Again, great questions. Thanks to everyone for sending them in, either Twitter, #ProudToBeND, via email, proudtobenotredame@gmail.com. We're also taking them via the Q&A bar if you're watching us on Google Hangouts right now. James asks, I'm considering a job in big data and bioinformatics. How much of a role does big data play in the research that either of you are involved with?
Let me jump in there. The proteomic studies that we do and the proteomic studies that my colleagues do and associated with the sepsis project generate huge piles of data. We run instruments for 24 hours a day for 5, 6, 7, 8 days, generating terabytes worth of data. And simply processing through all of that data, sifting through all of that data for the few needles that characterize the disease and outcome of sepsis is a huge undertaking. Then you multiply that by the large number of patients that need to be studied, characterized. It becomes a significant challenge. So I congratulate you in a career choice in choosing bioinformatics and big data, because that field is in desperate need of more researchers and talented researchers. You'll have good employment opportunities.
Another question we have, Stella has asked us, this type of genetic medicine and information might have consequences outside of medicine. Should a patient know if he or she is likely to develop a disease with no treatment or cure? So likelihood of disease, the markers that say, yes, I'm susceptible. We know there are some tests already that can point to certain predilections for diseases. How does the work that you do influence that kind of predictive modeling, and what are your thoughts on kind of the field in general?
We had to face this in the Human Genome Project, and the U.S. government's human genome programs were very sophisticated and devoted several percent of the overall budget to dealing with bioethical issues. And this was a key one. And the studies that I can recall dealt with Huntington's disease. That's the devastating disease which neurological degeneration and death in the 40s and 50s. And as I recall, when the population was asked, do you want this information? Not that this information will lead to improved therapy. This information will allow you to choose how give you information on your likely lifespan and the onset of this disease. And it is my recollection, my understanding, that the population was very much interested in having access to that information. It was subtle. Some patients or some individuals who were at risk for Huntington's may have had parents who died from the disease. When they found out that they didn't carry the gene, that they were going to be disease-free, felt emotional letdown. And others who found out that they did carry the gene for the disease, and were likely to get the disease, were able to arrange their life in such a fashion as to maximize the pleasure. It's a very subtle, challenging, interesting question, and we have now about 15 years worth of experience for many of these diseases. It would be interesting to revisit the literature and to see how the bioethicists are finding the populations, but facing the questions that you ask.
Great. Well, we're looking to wrap up our time here. I wanted to finish with a question kind of for the both of you and let you answer. The work that you do individually, how does it best reflect the greater mission of Notre Dame, that need to create forces for good in a world deeply in need? How does Notre Dame's mission affect your work, and how do you continue to work through that in your day-to-day research? Professor Bohn, let's start with you.
Certainly. Well, I think Father Jenkins has been very clear that when he thinks about Notre Dame, he really thinks about the three pillars of our mission being undergraduate education, research and scholarship, and our unwavering dedication to our Catholic identity. And I would say that I think that the precision medicine program and the work that we do really hits all three of those. Norm has already talked about the important role that undergraduates can play in this research, and we both work with undergraduate students in a variety of guises, but especially within the precision medicine program in our own individual laboratories. And undergrads they're just terrific. If I had to compete for a job against Notre Dame undergrads, I'd probably be, running a gas station somewhere or something. They're just terrific young men and women. With respect to research and scholarship, I think the answer there is obvious. This is a wonderful training ground for advanced students, both doctoral students and postdoctoral students who are working in our laboratories, because they're getting to work on the cutting edge of technology and science. And then I think most importantly, and one that really distinguishes Notre Dame from our colleagues at other universities, and that is we have a specific vision of a university which focuses on, as one of its tenets, a dedication to social justice. And so as we look at the work that we do, impacting people's lives, making it possible for people to have better treatments for illnesses. We very much think that fits nicely within the social justice understanding of our Catholic character as an institution.
Terrific. Norm?
Paul's really good and has articulated well. I'm going to go a little bit on the research and scholarship. The University of Notre Dame is a very unusual institution in North America, in the world, and the resources that we have available here and the commitment to developing strength in a number of research areas is really quite exciting. And I think that the opportunity to develop a strong program in analytical chemistry and sciences in general, and in precision medicine in particular, is a very exciting opportunity for us as researchers and for the university as a whole.
Much. Thanks again to all of you for joining us today in our first Join the Fight webinar. If you'd like to join the fight, I invite you all to go on donate.nd.edu, and when you make a gift, you can enter in the text field, you can enter Fighting for Precision Medicine to support the amazing work of these two men, Professor Norm Dovichi, Professor Paul Bohn. Thanks again so much for all that you do. Thanks again to everyone for watching us. We'll see you very soon here with another Proud to Be ND Join the Fight webinar, and go Irish. Thanks again for joining us.
Thank you all.